Karlo is thermal pace hissing poramil. Photosynthesis is arguably the most important biochemical process on the planet. It's how plants capture light energy and convert it into chemical energy, ultimately feeding nearly every food chain on Earth. In this video, we'll break down where photosynthesis happens, the light reactions, the Calvin cycle, and the special adaptations of C4 and CAM plants. This is one of the highest yield chapters for NEET, so let's get into it properly. Photosynthesis takes place inside chloroplasts, organelles containing stacks of membrane discs called thylakoids, arranged into structures called grana, all surrounded by a fluid matrix called the stroma. This physical structure directly maps onto the two stages of photosynthesis. The light reactions, also called the light-dependent reactions, occur on the thylakoid membrane, where chlorophyll and other pigments absorb light energy. The Calvin cycle, also called the light-independent or dark reactions, occurs in the stroma. Despite the name, the Calvin cycle doesn't create darkness. It simply doesn't directly use light itself, but it does depend entirely on the ATP and NADPH produced by the light reactions. This is a critical exam point. If you stop the light reactions, the Calvin cycle stops too, even in continued darkness, because it runs out its energy supply. The light reactions begin when chlorophyll and accessory pigments, organized into clusters called photosystems, absorb photons of light. Photosystem 2, despite the confusing numbering, acts first. It absorbs light and uses that energy to split water molecules in a process called photolysis, releasing oxygen as a byproduct. This is where all the oxygen you breathe ultimately comes from. The excited electrons from photosystem 2 travel through an electron transport chain, losing energy along the way, which is used to pump protons across the thylakoid membrane, building up a concentration gradient. These electrons eventually reach photosystem 1, which re-energizes them using more absorbed light, and they're finally used to reduce NADP plus into NADPH. Meanwhile, the proton gradient built up across the membrane drives protons back through an enzyme called ATP synthase, and this flow powers the synthesis of ATP from ADP, a process called photophosphorylation. So the two key products of the light reactions are ATP and NADPH, plus oxygen as a byproduct. The Calvin cycle uses the ATP and NADPH from the light reactions to convert carbon dioxide into sugar, and it happens in three phases. In the carboxylation phase, the enzyme Rubisco, the most abundant enzyme on Earth, attaches a molecule of CO2 to a 5-carbon acceptor called RUBP, forming an unstable 6-carbon compound that immediately splits into two molecules of a 3-carbon compound called 3-phosphoglyceric acid, or 3-PGA. In the reduction phase, ATP and NADPH are used to convert 3-PGA into G3P, a 3-carbon sugar, which is the actual usable product of the cycle. Some G3P molecules leave the cycle to build glucose and other larger molecules. In the regeneration phase, the remaining G3P molecules are used with more ATP to regenerate RUBP, so the cycle can continue. To make one net molecule of glucose, the cycle must turn 6 times, fixing 6 CO2 molecules, using a total of 18 ATP and 12 NADPH. These numbers are commonly asked directly in exams. In hot highlight N photosyn runs, Karlo is thermal parasyne with oxygen instead of carbon dioxide, in a wasteful process called photorespiration, which reduces the efficiency of sugar production. C4 plants, like maize and sugarcane, evolved a clever workaround. Their leaves show a special arrangement called Kranz anatomy, where the vascular bundles are surrounded by a ring of large bundle sheath cells, which are in turn surrounded by mesophyll cells. In the mesophyll cells, CO2 is first captured not by Rubisco, but by an enzyme called PEP carboxylase, which has no attraction to oxygen at all, forming a 4-carbon compound, which is why it's called the C4 pathway. This 4-carbon compound is then transported into the bundle sheath cells and broken down, releasing a concentrated burst of CO2 right where Rubisco and the ordinary Calvin cycle are located. This concentration effectively suppresses photorespiration, giving C4 plants a real efficiency advantage in hot climates. The rate of photosynthesis depends on several environmental factors, and understanding how they interact is a common exam theme, based on what's called the law of limiting factors. This law states that when a process depends on multiple factors, its rate is limited by whichever factor is closest to its minimum value at that moment, regardless of how favorable the other factors are. Light intensity increases the rate of photosynthesis up to a point after which the rate plateaus because some other factor, often CO2 concentration, becomes limiting. Carbon dioxide concentration works similarly. Increasing it boosts the rate up to a saturation point. Temperature affects the rate through its influence on the enzymes involved, including rubisco, with an optimum temperature range beyond which enzyme activity and therefore the rate of photosynthesis actually declines. This means that on a cloudy, cool day, light might be the limiting factor, while on a bright warm day with normal atmospheric CO2 carbon dioxide concentration often becomes the limiting factor instead. Let's recap. Photosynthesis happens in two coupled stages. Light reactions on the thylakoid membrane produce ATP, NADPH, and oxygen, while the Calvin cycle in the stroma uses that ATP and NADPH to fix carbon dioxide into sugar via rubisco. It takes 6 turns of the Calvin cycle use 18 ATP and 12 NADPH to build one glucose molecule. C4 plants use Kranz anatomy and PEP carboxylase to concentrate CO2 around rubisco, minimizing wasteful photorespiration in hot climates, and the overall rate of photosynthesis is governed by the law of limiting factors, where light, CO2 concentration, and temperature can each become the bottleneck depending on conditions. That's photosynthesis in higher plants. This chapter connects deeply to cellular respiration and plant physiology, so make sure these concepts are solid. See you in the next video.